My name is Courtney Gamston. I work at the Johns Hopkins Bloomberg School of Public Health in the laboratory of Dr. Jason Rasgon. Today we'll be doing bacterial purification from cultured insect cells. It's actually a very straightforward procedure. We start by harvesting our cells and then physically disrupting the eukaryotic host cell membranes using glass bead lysis. And then we use differential centrifugation to separate the bacteria from the host cell debris. And then we use a series of filtration steps to further remove host cell particles so that at the end we are left with just the purified bacterial pellet. This is actually one of the major requirements. It's a cell culture hood, and it's used to keep everything sterile because cell culture is really easy to contaminate. We also need cell culture media, the cells themselves, and we grow them in plastic flasks. We also need 3mm glass beads, 50 ml conical tubes, and we need a centrifuge that is capable of a high-speed centrifugation, approximately 18,000 G to pellet our bacteria, but also capable of doing about a 3,000 G or 2,500 G to pellet our host cells. So the first thing we'll do is we'll start with our cultured cells. They're grown, these are T75 flasks. They have 15 mLs of culture media as well as our cells. So the first thing we'll do is start by disrupting the cell layer and collecting them in a 50 ml centrifuge tube.